Dr. Lee Murphy, Ashton Ralph, Etienne Legge, and Yvonne Mayel are 4 leading scientists in breast cancer research that have helped to establish not only the Manitoba Breast Tumour Bank, but clinical trials and have evolved a lot of the community in different hospitals. So it's not just cancer care, but we get samples sent to us from all over different hospitals in Manitoba, and even some of the plastic surgery centers, because we also, not only do we study cancer, but we need to know what cancer isn't. And so when we need to look at normal breast tissue, women that are undergoing voluntary breast reduction surgery can donate their tissue to us. It goes into the Tumour Bank, and we now have a control group. So these are women that have normal breast tissue, and we can compare what their tissue looks like to the tumor tissue. And the tumor tissue that comes in here in the Breast Tumour Bank is part of a larger network across Canada. The Canadian Tumour Repository Network, or CTRNet, is a collection of tumor banks located in BC, Alberta, Ontario, and Quebec that we're all linked up with using a shared database. And if I'm a researcher and I need to collect, say, 40 tumors from premenopausal women with HER2-positive tumors that are, that haven't undergone chemotherapy yet, I mean, that's a really specific, defined group of women. It's going to take me a long time if I'm just going to wait and recruit women from Manitoba. But if I can get 6 tumors from BC and 5 from Ontario and a couple from Quebec, my research project can hit the ground running in a matter of weeks after my protocol's been approved, instead of having to wait years to try and get as many as I possibly can. So the advantage of a tumor bank, much like having everybody in the same building, we have all these tissues. They're all in a database. We know how old the patients were, their medical history, what they were given, who treated them. We have access to all that information, and we have access to it across Canada, and we can share that between places. So one of the things I wanted to show you is when we donate a tumor, I mean, we all have these images in our head that the tumors are big and gigantic and ugly and horrible, but this is actually somebody's breast tumor. It's kind of tiny. We have an agreement with Diagnostic Services Manitoba, it's that little yellow dot right in the middle there, with Diagnostic Services Manitoba, that if any woman or man who has a tumor that's greater than 2.5 centimeters is having the tumor removed, that we can get a portion of it. So this is all we get, and it's really, really important for the research purposes. But if I cut off, if I shave off the top and do analysis of that, and then the next week I come back and shave off a little bit more, I'm going to run out. So how do I compare a lot of people all at once? Well, we take a little core sample from this tumor, and we put it into what's called a tissue microarray. So you can see it kind of looks like a bingo board, and each one of those black dots is a different tumor from a different patient. And we know where the position of every one of these tumors, who the people were, what they had done, what the results were, what the pathologist said. And so when I cut off a section at the top here, I can then say, okay, the person at row 3, column 2, those results are different than the person in row 4, column 1. So we have a tissue microarray that can screen over 60 patients at once, instead of doing them one at a time. And we take out these little tiny cores and we make a grid like this, so that when we're using as much tissue as we can as efficiently as possible. Because this tumor represents a point in time that we can never go back to. So when we have something like this, we can do more analysis faster. So the point of the tumor bank is to make as many of these as possible using as many different types of tumors. So we collect lung, ovarian, prostate, head and neck, breast, obviously the breast is the big one, as well as blood and urine samples from people who have leukemia or any of the other types of blood disorders. But we also collect blood and urine samples from people with other types of cancer. So one of AFSHIM's projects is looking for circulating tumor cells. And these are cells that have broken away from the tumor and are migrating through the body to spread. So if we can try and isolate those cells and identify them in the blood first, before they go over from, like they leave the breast and go to the liver, or they go from the lung and embed themselves somewhere else, like in the colon. You don't want metastasizing cancer. So if we can identify those circulating tumor cells from the blood of people who we know have one type of cancer, maybe we can help them before they get another type of cancer. So that's the goal of the Tumour Bank.